Assalamualaikum warahmatullahi wabarakatuh, and a very good day to everyone. So this is a briefing for lab 2, and for this lab 2 we're going to do on a gram staining only. So before we do the gram staining experiment in the lab, so I think I have to give you an introduction on what gram staining is about and why do we want to do it. Okay, so basically gram staining ni adalah suatu method yang secara preliminary ataupun the initial method yang kita guna to identify or to classify the bacteria into different groups, two big groups. Okay, so the first group will be the gram positive bacteria, and the second group will be the gram negative bacteria. Okay, so the differences between the gram positive and the gram negative bacteria, it lies on these two different structures, okay, on those gram positive and negative bacteria. So the first one is on the peptidoglycan thickness, and the second one is the presence of the outer membrane. Okay, so we can see here for the gram positive and gram negative, they both have the peptidoglycan layer. Okay, but the thickness is different. So for gram positive, they have a very thick peptidoglycan layer, but for gram negative, they have a thin, peptidoglycan layer. So that's one difference. The second difference of gram positive and gram negative is the availability or the presence of the outer membrane. Okay, so if you can see here, you can compare the gram positive and also the gram negative. The gram negative, it has the outer membrane, but not for the gram positive. Okay, so these two structures in the gram positive and gram negative, it will determine in the gram staining process, and that will show us later in the lab on how to identify whether the sample that we have, the bacterial sample that we have, it contains the gram positive or the gram negative bacteria. So this is the mnemonics on how to remember how to identify the gram positive bacteria and how to identify the gram negative bacteria. Okay, so at the end of the gram staining process, the smear or the cultures will only be with two colors only. So it will be either purple or something reddish or pink. Okay, so if purple color, it means that that bacteria is a gram positive bacteria. So how to remember? Positive, it has P in it, and P stands for purple. Okay, so remember that. And the other one, gram negative, it will be in the reddish pink color. Okay, so this is how for you to remember. This is the process, okay, for gram staining. So let's say there are 1, 2, 3, 4, 5. So there are 5 steps. So the first one is the fixation. So meaning the fixation is you will put the culture, the bacterial culture, onto your glass slides, and you will fix it and waving it through the fire. Okay, and then the next one is the crystal violet. Then you will put in the crystal violet solution. And then from the name itself, because it has the color of the violet color, then at that time we will not see the differences between the gram positive and gram negative. They will be in violet or blue color. Then the next one is the iodine treatment, where we put the iodine solution, and at this time still we won't see the difference between the gram positive and the gram negative bacteria, because both will take up the color of the iodine, which is purplish. The next step is the decolorization, and the decolorization, at this point we will see the difference between the gram positive and the gram negative, where the gram positive, it will stay color purple. okay, and gram negative, it will be decolorized by the solution that we use in this step of decolorization. And the next one, the last step, is we will counter stain with the safranin solution. So safranin solution, the color of that safranin is reddish pink. So at this point, it will tell us strongly that gram positive is the one with the purple color, and the gram negative bacteria is the one with the reddish pink color. Okay, so this is the steps and the timing. Okay, so the first one is we will have the smear, and then we will put the crystal violet first. Okay, remember, so the crystal violet, it has the color of purple or blue. Okay, and at this point, all of our bacterial cultures will be stained with this crystal violet, so we won't see any difference yet. And the next one is for the iodine treatment. Then iodine treatment, still no color changes yet. Then the decolorization step. So usually for the decolorization step, we will use ethyl alcohol, or we can use acetone or any alcohol. And at this point, we can see here differences in the color, where the gram positive will remain the color of purple, and the gram negative will be decolorized. And the next one, when we stain with the safranin, which has the color of reddish pink, then the gram negative will take up the color of the safranin, and from that we know that those are the gram negative. Okay, so we will do this step in next week lab. Okay, that's all. Thank you and all the best.